In this section, we're going to talk about the experiment of Frederick Griffith, that in 1928, he proposed the transformation principle using as a model organism a type of bacteria that produce pneumonia, and he also used mice. Now I'm going to talk about how Frederick Griffith discovered bacterial transformation, and I think it's very relevant to what is happening right now to talk about the Spanish flu, because similar to the coronavirus, we have a flu epidemic, and it was so detrimental to all humanity that government rushed to develop vaccines that could stop the spread of infectious diseases. And I'm going to tell you a little bit more about this pandemic. So the Spanish flu was an unusually deadly influenza pandemic, different from the coronavirus. That one infected 500 million people, and the death toll is estimated to have been anywhere from 17 million to 100 million. And why there were so many people infected, and this is a lesson that we can learn, and actually we'll learn from the past, that in that case they minimize the early report to maintain the morale of the people. Yeah, so it was the First World War, and to maintain the morale of the people involved in the war, censors minimize early report of both illness and mortality in countries like Germany, United Kingdom, France, and United States. It was They were so minimized the early reports that we still don't know where the Spanish flu originate. Yeah, we don't know. Some people think that it was in Kansas in the United States, or people think that it's in China, that it originated in China. We just know that it didn't originate in Spain. Actually, as Spain was not involved in the First World War, he was able to journalists start reporting about this very atypical pandemic, the atypical influence, that it was very deadly. So again, I think we learned very well the lessons from the past, and so now we know that being far from each other is very important to prevent people to get sick. And now I'm going to go back to the work of Frederick Griffith. Now, as I said before, the governments of all the world rushed to develop vaccines, and many scientists were working on pneumonia, in particular a bacteria that causes pneumonia, that is an inflammatory condition of the lung affecting primarily the small air sacs. And one of those scientists was Frederick Griffith, that he was working in England, and he was a microbiologist. And the key component of this experiment was that he was working with two strains of Streptococcus pneumonia. So the strains of the bacteria have two different phenotypes. One phenotype is that, so we have strain R and the strain S. R means rough and S means smooth. The rough means that the colony morphology is rough, and the S means that the colony is smooth. And the other phenotypic difference is the virulence. One, the rough will not be virulent, and the smooth will be very virulent. So Streptococcus pneumonia vary dramatically in both their appearance and virulence, and their ability to cause diseases. The highly virulent S strain had a smooth capsule, as you can see in this schematic. Here you have the S strain, and here you have the R strain. That is very important because it allowed them to identify easily the bacteria. And the S strain has a smooth capsule or an outer coat composed of polysaccharides. On the other hand, the non-virulent, the R strain, had a rough appearance, lack of that capsule. And those were key components of the experiments. Now I'm going to talk about the experiment and how what is happening that he talked about transformation between bacteria. So in the beginning, he took bacteria that it was virulent and injected to mice. So he found this bacteria that smooth cells, and after a few days, the mouse contracted pneumonia and unfortunately died. But when he took the mice body and he is able to isolate S colonies from the tissue of the dead mice. So here you can see in a petri dish the the colonies of the virulent bacteria, and in that way he confirmed that the mice was killed by the bacteria. Then he took the non-virulent, the rough uh, appearance bacteria, and when he injected to the mice, as he expected, the mouse remained healthy, and he was able to confirm these results by isolating R colonies from the tissue of the mice that it was alive. And the next experiment that he did is he used heat to kill the bacteria that it was virulent, yeah. And when he killed those bacteria, I remind you that bacteria has, similar to us, has proteins, and those proteins need to have a limit. Yeah, if the temperature increases too much, those proteins are not functional anymore. This is why you have fever, to try to kill the bacteria. And in that case, he killed the bacteria using heat. And when he injected the bacteria was not alive anymore, and he tested that by culturing again the bacteria he took from the body of the mice that he was alive. And he tried to culture the bacteria, and he didn't find anything. So he found three key experiments. And so far, he has done three key experiments, injecting the bacteria with one that is virulent, one that is non-virulent, and one that the bacteria was virulent but it was dead. And now he combined the two previous experiments, one that it was non-virulent, and he mixed it with one that it was dead but it was virulent. And he found something that it was very interesting. When he injected to the mice, he found that the living R cells plus the heated kill S cells were able to kill the mice, yeah. And when he isolated the cells from the body of the mice, he found that both R and S colony were isolated from the tissue. Yeah. So while here he didn't find anything, here he found both. And what are the implications of that results? He found that heat kill S cells can transform the R cells, making it virulent. Yeah. So the ones that they were non-virulent when they interact with the one that they were the non-virulent when they interact with the dead ones that they were virulent, both they can interact and they transform each other, and now the mice were dead. Yeah. So as a conclusion, he concluded the genetic material of the S cells can be transferred to an R cells, resulting in both virulence and the production of a smooth. And now I'm going to tell you a little bit more about what we know about this process. Yeah, what we know about the presence of these experiments. Well, now we know that bacteria display many mechanisms to transfer genes from one bacteria to the other one. Some of them called conjugation, transformation, and transduction. Yeah, and I'm going to talk a little bit about that. And today we know that the DNA molecules can renaturate after heat treatment. So similar to the proteins, the DNA also separate the two strains. They break the hydrogen's bonds and break, and they separate. So instead of having the double helix, you have two strains separated, but after they cool down, they can come back together. The bacteria can take up foreign DNA from the environment by a process that we still call transformation. So let's talk a little bit about that. So as I said before, there are mechanisms to transfer genes between bacteria, and we're mainly going to focus about one called transformation. And here in this schematic, you can see that bacteria, and here in green you can see a bacterial exchange DNA, so from a different environment, from a different bacteria. And here you can see the chromosome, the circular chromosome of a bacteria. Bacteria are less complex than eukaryotic, so instead of having linear chromosome, they have circular chromosome. And the important phenomenon, the important mechanism that is happening here is that the DNA that comes from outside of the cell, from this bacteria, it can attach to the bacteria to this process called transformation. So the transformation experiment, it was, now we know what is happening. The bacteria that they were virulent, that they were dead, they can be, that DNA can be attached to the other bacteria, and they can be part of the other bacteria. So what is bacterial transformation? Now we know in the present that we define bacterial transformation as the generic alteration of a cell resulting from a direct uptake of DNA. And how it happened, there is an incorporation of exogenous genetic material from its surrounding through the cell membrane. So it's very interesting, although at that time he didn't know what was the transformation principle. He didn't know if it was DNA, proteins, lipids. He just know that something was causing the change. Now we know that the transformation principle, it was because one bacteria was absorbing, was taking the DNA from another bacteria. And this is a process that is very important because bacteria can easily get antibiotic resistant by sharing multiple genes. And I've talked about the transformation, but still there are many more. In the next section, we will talk about Avery and his colleagues that in 1944 found that the DNA is the transformation principle. And again, by using a similar experiment, he used now proteins called enzymes that it will allow them to find that DNA, not proteins or RNA, is the transformation principle. So, as I said before, inspired on the work of Griffith, Avery in 1944 and his colleague provide the first evidence that DNA was the genetic material. Well, so they link what is the transformation principle? The transformation principle is DNA. So the scientists identified the chemical substance responsible for changing the rough non-virulent bacteria or non-virulent cells into the smooth encapsulated infection cells. And I'm just going to tell you again what is the main conclusion, and then I'm going to tell you how they came into this conclusion. They found that the transformation factor that turned these R cells into the smooth cells, it was the DNA. It was not the RNA, it was not the proteins, it was not other molecules, it was mainly the DNA. And to arrive to these conclusions, they did a process of elimination to identify the transformation principle. And how they did that, there is a type of proteins, as I said before, they are called enzymes. And those enzymes are going to break into pieces, for example, proteins, or they are going to break into pieces the RNA. So the enzyme that break proteins are called protease, and the ones that break the RNA are called RNase. Yeah. So what they did is they took the bacteria with the transforming activity, and they know that the transformation principle, the transformation activity, even if you heat the bacteria, the transformation principle is still going to work. So what they did is they heat up again the S bacteria, and what they did is through the elimination. So they use the enzymes with RNA, the enzymes with protease that they will break. This one will break the RNA, this one will break the protease, and this one will break the DNAs, will break the DNA, and in that way they can judge, they can break the molecule that it was in charge of the transformation principle. And if they break it, they will not see a transformation. So let me walk you through the experiment. So here they have the S cell extract. The cells were previously heated, so they are dead, but the transformation activity still take place. So when they are the RNA that RNases that destroy the RNA, here they did a similar experiment with an enzyme called protease, and again this extract has dead cells, and they culture it with our cells. Yeah, so they are not, if the transformation principle was destroyed, it was RNA or it was RNA or it was proteins. It was not supposed to be a transformation from S to R. However, they found that they found both type of cells. They found that there were R colonies, as they expect that they culture, but they also found S colonies. Yeah, so they found R colonies and S colonies in both cases. So there was, to some degree, some amount of transformation. When you heat up the bacteria, you kill them, and you put RNAs, aces, or proteases. So it means that these two, the RNA and the proteins, they were not the transformation principle. On the other hand, when they add the DNA into the bacteria that they were dead, and they cultured it with the R cells, what they found is, if you see here in this schematic, you see that while here you find the two types of bacteria, here you only find one. You find the R cells, you don't find the S cells. So it means that the transforming activity was destroyed by the DNAs, but not by the RNAs or the protease. In this way, showing you that it was the DNA, the molecule, producing the transformation principle. So far, based on this experiment that I just explained to you, the genetic material can be transferred from one bacterial cell to another one. However, there is still one question. Can DNA be transferred between generations? And this question was answered by the next group of scientists. So the next group of scientists, the next piece of information, was found by Hershey and Chase in 1952, and they found that the DNA is the generic material of the T2 phage. And now, to summarize all the information that we have seen, I'm going to show you a video that it will talk about these key points that I just explained.
Then in 1944, out of left field, came an experiment that changed the game.
Avery, McLeod, and McCarty did an eloquent experiment with the bacterium pneumococcus.
The bacteria that cause pneumonia. There were different strains of the bacteria, and one of those strains was virulent, meaning it caused the disease. Then you had another form of the bacteria that was non-virulent.
Oswald Avery showed that you could convert the benign form into the virulent form when the only thing exchanged was DNA.
These bacteria, when they divided, their descendants could be killers too.
This, therefore, was definitive evidence that DNA was the molecule of heredity.